Imagine you are developing a new polymer for a medical device or optimizing a coating for better durability. Everything looks perfect until performance testing fails. And the reason? Something as fundamental as molecular weight distribution. That's where Gel Permeation Chromatography, or GPC, becomes critical. At Resolve Mass Laboratories, GPC is not just a test. It's a powerful decision-making tool that helps industries across North America understand their materials at a molecular level. Today, I'll walk you through not just what GPC is, but why it truly matters in real-world polymer characterization. Gel Permeation Chromatography, also known as Size Exclusion Chromatography or SEC, separates molecules based on their hydrodynamic size in solution. Not their chemistry, not their charge, but their physical size in a solvent environment. In simple terms, larger molecules move faster through the column because they cannot enter the tiny pores of the stationary phase. Smaller molecules, however, enter these pores and take a longer, more tortuous path. This difference in travel time allows us to generate a molecular weight distribution profile, which is far more informative than just a single average molecular weight number. In polymer science, knowing only one molecular weight value is like knowing only the average income of a city. It doesn't tell you the full story. GPC gives us MN, MW, and MZ, number average, weight average, and Z average molecular weights, each offering a different statistical perspective. More importantly, we calculate the Polydispersity index, or PDI, which is the ratio of MW to MN. A PDI close to 1 indicates a very uniform polymer population, which is ideal for high-performance applications like drug delivery systems. Higher PDI values suggest broader distributions, which can impact mechanical strength, viscosity, processing behavior, and regulatory compliance. Let's visualize what happens inside a GPC column. The column is packed with porous gel particles. When your polymer solution enters the column, molecules distribute themselves between the pore volume and the interstitial space. Large molecules are excluded from most pores and elute first. Medium-sized molecules partially penetrate pores. Small molecules enter many pores and therefore elute last. In practical laboratory terms, this means retention time directly correlates with molecular size, but only after proper calibration using standards and validation of system suitability parameters like resolution and reproducibility. In the polymer and plastics industry, GPC is essential for determining molecular weight averages and understanding branching or cross-linking, which directly affects tensile strength and processing performance. In pharmaceuticals and biotechnology, GPC is critical for characterizing drug delivery polymers, pegylated molecules, and even certain biomacromolecules. Regulatory agencies expect thorough molecular weight profiling for consistency and safety. Similarly, coatings, adhesives, environmental science, and academic research rely on GPC to evaluate material stability, degradation behavior, and long-term durability. At Resolve Mass Laboratories, GPC services go beyond basic molecular weight determination. We provide comprehensive analysis, including MN, MW, MZ, PDI calculation, polymer structure evaluation, and quality control testing aligned with industry standards. One of the most valuable services we provide is method development. Not all polymers behave the same. Solvent compatibility, column selection, detector choice, RRI, UV, or multi-angle light scattering, and calibration strategy must be optimized. Our experts ensure that each method is tailored to the specific chemistry and application of the sample. High-quality GPC analysis depends heavily on instrument precision, calibration integrity, and scientific expertise. State-of-the-art systems ensure reproducibility, but it's the experience of analytical scientists that ensures meaningful interpretation. We focus not just on generating chromatograms, but on translating data into actionable insights. For example, if molecular weight drift is observed between batches, we help identify whether the issue is due to polymerization kinetics, degradation, or processing conditions, enabling real corrective actions. Resolve Mass serves clients across Canada and the United States, supporting industries ranging from pharmaceuticals and medical devices to packaging and advanced materials research. Our multidisciplinary team combines expertise in polymer science, analytical chemistry, and materials engineering. Beyond GPC, we integrate complementary techniques such as FTIR, DSC, TGA, NMR, and rheology, because true material characterization often requires a multi-technique approach for complete understanding. If you are developing a new material, troubleshooting batch inconsistencies, or preparing documentation for regulatory submission, GPC analysis can provide the molecular-level clarity you need. At Resolve Mass Laboratories, we offer technical consultation, customized method development, and detailed reporting designed to support your R&D, quality control, and compliance objectives. We don't just run samples, we help you understand them,